The talk is by Dr. George C. John, Advances in Imaging in Renal Pathology.
Is this a nice time to talk? This talk will be different because you don't have to remember anything. Just feel the talk. Because preparing it, I realized what's written here by the famous balladeer singer Leonard Cohen, whose songs are there on the net. Anna Wilson is familiar, and the lyrics are beautiful. While I organized this talk, I realized this cannot be a perfect offering, but that's, I believe, its merit. Through the cracks, I hope light will shine. I have got here visualization in nephropathology, and I know none of you can read it, because that is where we are with nephropathology. And this can be understood only from different dimensions. I hope you will relate to at least some dimensions that I have here in these beautiful space-friendly octagons. Imaging in human experience, a few slides, something about ultrastructure of the kidney, digital pathology, virtual microscopy, deep learning, computational nephropathology. These are words that you will hear and which will have meaning as we go on. And 10 years later, you will say, now probably it makes sense if you ever remember it. Detection of new entities from old pathological processes that we understand, are familiar with. And what are the new technologies? There are quite a few mentioned here. I won't read it out for you. Then finally, where do they converge? Is there an integration? And what is genomic GN? And finally, let me conclude with some nephropathologists who we must remember of Indian origin. This particular lens is kept in the British Museum, 3,000 years old at least. One of our ancestors must be 10, 12 generations ago must have peered through it. It is about 11 centimeters in size, made of rock crystal. It's got a magnifying factor of 4. So that person must have seen something that was fascinating, something enlarged. And that's where recorded quest of human visualization started, I believe. We have radio telescopes all around the world, and there was this project that summoned up all 5 or 6 radio telescopes all around the planet, focusing on a certain area in the cosmos. At the center of the galaxy M87, collecting data for 5 consecutive nights 2 years ago. And the picture that emerged from that was published in April this year, 2019. And they called it Powehi. This is the first picture of a dark black hole that has ever been made. It was there somewhere 55 million years ago. We do not know what shape and size it is now, or if it exists. And it was at least 6.5 billion times as large as our sun. So from the very large, through quantum computing that has become reality now, quantum supremacy, which meant that the current ability of a quantum computer, a super cool chip, to process information in 3 days, what a current supercomputer will take 10,000 years to do. That means the supremacy is clearly established. This is a paper published in Nature. You can see Sundar Pichai's face through that picture. and further down, delve deep, deep, deep into matter. What do we find? Using cryogenic scanning tunneling microscope. Here are the pictures of titanium atoms. An atom can be pictured today. And this again has happened this year. So there is something magical about 2019. There's something magical about this time. Now coming to the kidney. Regular microscopy, electron microscopy, Wello had one of the first ever in this country for medical purposes. Dr. C.K. Job, I remember mentioning that leprosy, Karigiri had the first electron microscope. We subsequently got one through Wellcome Foundation, which is still available for us for clinical use. And this is one step further forward using helium ion. And look at the clarity of this glomerulus. I don't know whether this is seen with so much of light around. The glomerulus and its structure, rather like a tiny little cabbage. And if you peel that away, you would find a cup, which is actually the Bowman's capsule. And if you look carefully on the surface, further magnified, you can see interdigitating processes that we talk about. And even further, that these processes that interdigitate further have interdigitating processes which I have never ever seen before searching for information for this particular talk. These cartoons you are familiar with derived from those pictures that you've seen, that when you chop a glomerulus like you cut a coconut, you find the afferent arteriole coming in, efferent arteriole leaving, breaking up into capillaries, the surface of which you saw earlier, covered by podocytes in yellow and endothelial cells in red, and blood flowing through these spaces here depicted white. And this tiny bit magnified is right here, showing two more elements. A mesangial cell which seems to coordinate the functions of capillary loops and the trafficking of molecules. Also, a basement membrane here in green, which forms an investment upon which sits the podocyte and underneath which sits the capillary endothelium. The structure is important as we go on because antigen-antibody complex formation gets deposited in different parts of this particular structure, the mesangial cell, the fenestrated endothelial cell, and the yellow epithelial cell with podocyte, the projections. And you can see here arrays of antibodies at different levels of engagement, some in the epimembranous region, above the membrane, under the podocyte, like in membranous nephropathy, some in the mesangial area, and some in the subendothelial region. Here's the helium ion microscopy, and here the standard scanning electron microscopy which we have here. Look at the difference in clarity of the same thing. This is the rat kidney, but you can see secondary processes of podocytes which otherwise we don't get to see. That's using helium ion microscopy. And beautifully seen here is the adjacent endothelial cells with what is known as a tight junction and the fenestrae of the endothelium. The food process have become extremely complex and it's a nightmare for all exam-going students in nephrology, and a very important part of pediatric nephrology training because each of these molecules depicted here have mutations that lead to childhood nephrotic syndrome. These podocytes are anchored with integrins and various other molecules that again with mutations of which make them fall off. Again, shifting track. Virtual microscopy. This is available in different parts of the world. The whole slide can be scanned using very precise mechanisms of slide movement, and the scanned information can be stored and sent across to different parts of the world. So much so the slide then in the glass format, physical format, can be removed, and the scanned information can be seen in America or wherever else you wish an expert to look at and give you comments on. The similar technology is used by hematologists for various karyotyping and other studies, as well as in neurology. There was 10 years ago an attempt at getting one of those microscopes for our purposes. So whole slide imaging is possible. Telepathology sends it around. Multiple students can view the slides, and they're used in trials, teaching, and research in the US, while Scandinavians are usually one step ahead, and they use it clinically in both Netherlands and Sweden. Regular stuff. Now, from the University of Miami, under Dr. Laura Bassoni, there is a huge big effort at trying to digitize nephrotic syndrome in all forms and manner. You can see here that the segmental sclerosis, whether it's a tip lesion or some other lesion, can be quantified. Mesangial hypercellularity can be quantified, and so on and so forth. Everything, interstitial fibrosis, tubular atrophy, as you know, is the most important deterministic factor for prognosis, and that can be quantified as well. Electron microscopic images can be stored, quantified, and sent across for harmonization. So computational nephropathology has come of age. It's no longer something that we think will happen, and it's only a question of time, because collaborative efforts are increasing all around the world and even in our own country. Whether it's enumeration of glomeruli, number of events that you're interested in studying, and amount of observational data, digital morphometry of cortical interstitial volume, fractional interstitial area, how much is the tuft volume, what is the cortical density of glomeruli. All these tend to be prognostic factors in different conditions, not just looking at a slide and giving a guesstimate of disease. Podocyte, the cell that sits on top of a basement membrane, its number and its health matters in terms of prognosis of nephrotic conditions. And that has now become podometrics as a separate discipline in condition. Some years ago, we were able to show that IgA nephropathy is a progressive condition amongst Asians, and this has now attracted a lot of attention after Sushina has taken it forward with a multi-center international study. The kind of computation that I talked about would lend itself beautifully to studies such as this, where we compare our histology with histology from other places and understand better why our patients do badly, even with a condition like IgA nephropathy. Convergence again happens in computing. As you know, artificial intelligence has evolved into machine learning. That has evolved now into deep learning. Sounds almost mystical. And I think this gentleman is also fairly mystical. I think Geoffrey Hinton, if you read what he has written. Essentially, this is very intuitive. Our brain functions like this. There are input neurons and there are output neurons, and in between there are intercalated hidden neurons. We know that. Now, based on the input, one has output. This could be auditory, visual, or any sensory input, and you have an output from the brain. What characterizes our learning is that with each iteration, our response to an input causing an output keeps changing from previous experience. So each time we get better at it. So there is a back propagation algorithm that's mathematically possible that Dr. Hinton first talked about and published on. As you can see, any of these neural networks can have a back propagation algorithm that modifies subsequent response to the same stimulus, making it better and better. So you can see how well deep learning can be involved in scanning slides and getting better results. Again, shifting our octagons, detection of new diseases using the newer technology. As you know, this is a picture of amyloidosis, Congo red positive stuff, birefringence, and fibrils on electron microscopy scattered at random of a particular size. This is amyloid. Now, we know that you can get sugar in fibrillary form that we can buy in the bakeries. Here's protein in fibrillary form. There is amyloid. There are different other types of fibrillary deposits. Now, what is this made of? It is possible today to laser microdissect. Can you see this? This is a glomerulus that has been marked out. Using laser, you can cut this, remove it. Look, it's been catapulted into a tray. These two are cut, catapulted out. This is the remaining tissue, and the catapulted tissue is subjected to mass spectrometry. And then you chart it along a proteomic chart, and then find out which are the proteins that it best resembles. In this case, it's leukocyte cell-derived chemotaxin 2, otherwise called ALEC2 amyloidosis. They have done 4 sets of experiments at publication about 3 years ago or 4 years ago, and this happens to be ALEC2 amyloidosis confirmed by specific immunoperoxidase stains. Yet another condition, fibrillary glomerulonephritis, PAS positive, Congo red and silver negative, IgG4 positive, random distribution of bigger fibrils, a different entity but a deposit disease. Similar technology, doing mass spectrometry, one found that this is actually DNA JB9 protein. I mentioned this specifically for a reason, and I'll come to it later. Again, this is the proteomic chart, and here the protein that seems to be most representative of this material that you see with immunoperoxidase stain specific for DNA JB9. You know, polycystic kidney disease is the 5th commonest renal disorder, primary renal disorder, and we know that it is to do with the polycystin channels of the non-motile ciliary processes that are there in the tubular cells, primarily. This is the histological representation of polycystin 1 gene arrangement and PKD2. And as you can see, using cryo-electron microscopy, one can actually see these channels. You can see these channels at higher magnification here. These can be eluted, and the PKD1 and PKD2, the 2 important genes, can actually be mapped. Its protein structure studied. And using X-ray crystallography and atomic mapping, you are able to map these, and you can actually see the polycystin channels. It is to this level of refinement that you can take renal histology. Membranoproliferative GN is the last I want to discuss in this domain. This is a disorder that we always understood as an infection-related proliferative glomerulonephritis, antigen-antibody complex formation, and a very infiltrated bloodless, large glomerulus, usually leading to massive proteinuria, hematuria, and renal failure. It so happened that we have in this complex always found once a year or maybe twice a year in a large biopsy center like ours here, just isolated deposit of C3 on immunofluorescence and nothing else. No immune complex. We never knew what it was, and we thought this was actually a resolving process. But now we know better, because as this paper shows, this is an abnormality of complement because only complement is deposited. And abnormal activation of C3 component of complement activates C5. That leads on to the membrane attack complex that destroys the glomerulus. So it makes sense actually to block either C3 or C5. And people actually have tailored molecules to suit that. This is the picture of eculizumab epitope. Eculizumab blocks C5, this particular domain, MG7, and that picture is from X-ray crystallography. This molecule is available to all of us to use. Just as before I left my hospital in Australia, we had a request for this particular molecule for a lady who had C3GN, and we looked at the cost. When I convert it into Indian rupees, it costs 6 crores for a year. So most of us can't afford it, but those countries which can still pay for it and use it. This blocks the complement activation. So there is a pathway and there are picturizations, and we are now able to find treatment that's tailor-made for it. Now coming to a few techniques which you are likely to hear more about. This is a multi-immersion open top light sheet microscope. Sounds terrible, but multi-immersion because you can keep multiple specimens mounted in some sort of a liquid tray here, which is, can you see the top of it? And here is the illumination objective, and here is the collection objective. Light goes in, the reflections are captured. These are the two arms. This is feeding laser through the light, and motorized top microscopy stage can slowly rotate the specimen to have laser beam pass through, and the light is collected. This is going to take a few seconds because we were not able to load it well. And the picture previous. Yeah. Multi-stained kidney tissue looks. You would wonder what pathologists will have left to do, but this does add a definite phenomenal dimension to where the cells are co-located and what the pathology is likely to be. It's scanning the whole volume of tissue. Thank you for that. And another technology is augmented reality microscope that cancer specialists, cancer histopathologists are using, not to replace them, but to help them with things, particularly in lymphomas, particularly when cancer need to be quantitated. Here's what the person sees. And this is the area that the computer has marked out, an area of interest because of its deep learning prior. And this is how it looks. And then on, the pathologist concentrates his or her attention on that area rather than on area that does not have the pathology that they are looking for. So once you know what pathology, let's say it's a lymphomatous tissue, there are multiple lymph nodes, or it's a breast cancer with lymph node dissections, those lymph nodes can be scanned using this particular augmented reality microscope, and then their attention can be very focused on areas of interest. Now, having two molecules of interest in a tissue does not mean anything. Even if they exist close by, it could be a random chance that you may have found those molecules together. But as you can see here, how do you know this? Computers with its ability from past tissues and algorithms can actually count how often are these molecules by size and stain co-locating. Is it by chance? Is it by a random movement or presence? Or is it truly, as you can see here, present at a much higher ratio, indicating likely etiological association? And finally, the last technique, a single molecule localization microscopy is available for living cells. If you have a particular compound, like here, it's a fatty acid analog that is used to stain this particular yeast cell, you can see that actually you can make that out as being present in endoplasmic reticulum. Shifting again, all this information can be integrated in different countries, Europe, America, and in China. And these informations can be shared across networks to harmonize tissue. What about one single tissue from womb to tomb? It's possible too. If you were to look at clinical phenotypes right at the bottom, histology, protein cytokine profiling, transcriptional profiling, epigenetic signatures using proteomics, and genetic variation. Integrate these 7 signals and then define disease mechanisms, functional subtypes, and predict clinical outcomes. This is the grand plan with renal tissue today. Now finally, to the nephrologists whom we remember, Indian nephrologists. Manjari Venkatachalam wrote his first paper from Calcutta in '64, continues to work as one of the top people in pathology in University of Texas Health Science Center. Second, Dr. Paranjit Randhawa, whom some of us know, graduated from AIMS, now works at the Pittsburgh Starzl Institute, and he is the first one to describe BK virus infection and multiple other things, very important. Dr. Swarnalatha Gopalan, 1 year my junior, she works in Hyderabad and arguably runs country's largest nephropathological services. And lastly, Priya Alexander, one of our own. She was one of the people who described the second protein entity that I talked about in fibrillary glomerulonephritis. She works at the Mayo Clinic. Most important of all, and to whom I dedicate this talk, is Dr. Anand Date, published about brain abscess long ago, 69, and published till about 2009 when he finally worked in Muscat and Oman, Qaboos University. Look at I've just given a sample of papers that he has published. GN in leprosy, one of the first papers, and where exactly the deposits were, and glomerular pathology and electron microscopic study by 77. A good number of you may not have been born then. And then go on with snake bite, hemolytic uremic syndrome with Shigella dysentery in British Medical Journal, cortical necrosis with Dobie nut. I don't know how many of you will know. Dobies used to use a marking nut on clothes to put number. That's a toxic substance. And he described probably these 2 or 3 cases that there are of renal cortical necrosis of somebody who accidentally ingested it. Then on to filarial chyluria, hemolytic uremic syndrome, snake bite, renal transplant, terminal infections. This paper is quoted multiple times in NEJM onwards in multiple journals. And subsequently, many more transplant papers. Alternate steroids, almost bordering on clinical work. And subsequently, one of the largest papers, descriptions of biopsy reports of 2,827 patients in 1987 from this hospital, published in Quarterly Journal of Medicine. And then on, changing a primary GN profile in Transactions of Royal Society of Medicine, 1989. He retired from Caboose University and lives in Oxbridge, right next to the Oslerien Library, where he spends his time. So finally, we the peace prophets, and we practice in the 15th century art of divination, otherwise called uromancy. Large bubbles of urine seen in a matula, right here. This is again from the British Museum, 15th century. This nephrologist is looking at the urine, obviously of this woman's, probably predicting gender of her baby, or if it had small bubbles packed tightly together, an illness or probably death of a loved one, or if there were large bubbles spread far apart, probably it was predicting that she was coming into a lot of money. Thank you for saving us from uromancy. Thank you.